Hey everyone, we're going to wrap up our semester-long exploration of various concepts in biology with the nervous system. The nervous system is made up of a variety of cells, but one of the champions of the nervous system is a cell called a neuron. And a neuron looks something like this. So there is a part of the neuron which is called the cell body, and the cell body is the part that's the classic cell part is the way of I thinking about it. It's got the nucleus in it. It has a lot of the same types of organelles that other cells have. And in addition to the cell body, though, there are these unique features of a neuron that make it different than other cells. One of those is that it has these branches, which are called the dendrites. And the dendrites are like the receiving end. What you could think of is you could think of it as that they receive electrical chemical signals. In other words, they receive electricity, so to speak. Now, at the dendrites, there's this big tug of war that happens. There will be other neurons attached to these dendrites. Some will be charging them up. Others will be taking away the charge. And there's this tug of war between charges to try and ultimately either inhibit or initiate what's known as an action potential. And an action potential is basically a change in voltage that sweeps along the axon. And this is the way that these cells communicate. So this thing here is called the axon. It's this really long extension of a neuron, and it can vary in its length. It can be actually super long relative to the size of the cell. It could be several feet long, or it could be only a few micrometers long. There's a huge variance in neurons in your body. But in either case, this is like the wire of the cell. Even if we think about these cells forming somewhat like an electrical wire, this is the part that is the wire. And this is going to transmit a change in voltage that sweeps across here, known as an action potential. It will only do that if the tug of war is such at the dendrites that will then squeeze, there's some signals that will be stimulatory, others will be inhibitory. Those are all acting on the dendrites, and if the nature is such that the stimulatory signals win out in that tug of war, then it's like firing the trigger of a gun. It shoots the action potential down the axon. It will go all the way down then to these, which are the terminals. And then neurotransmitters will be released by the terminals, and then they usually are going to be synapsed with some other structure. So the neurotransmitters will be released. They'll go and bind to some other structure, and if it's that other structure is a neuron, then it will start the whole thing over again. Okay, it would be neurotransmitters that are released and binding to the dendrites, which are causing them to either be stimulatory or be inhibitory. And so you can see that this then just repeats. And so the way that I think about it is neurons are somewhat like dominoes. In the same way, you can push a domino down and it will start a chain reaction that goes from one end all the way to the other. That's how neurons are in a way. You can, if there's enough force, so to speak, to get one to fire, it will start a chain of reaction. And as long as the force that it's exerting on the subsequent domino or the subsequent neuron is strong enough, the neuron will send an action potential, and then it will transfer to the next one, and then to the next one, to the next one, and then it can spread out throughout a whole network of neurons to go to different places and do different things. All right, so that's a little bit about what we got going on here. Now, there is something that's required to prime neurons so that they can respond to electrochemical signals. They can respond to neurotransmitters binding it, and it can either charge them up or discharge them. And the reason why they are able to respond in this unique way is that they have what's known as a resting membrane potential. Now, this word potential, it just means voltage, okay? In the same way that there is, let's say, the potential for you to create energy and do something by plugging in a cord into an outlet, there's also potential energy that exists across the membrane of a neuron. The definition of voltage is the potential for electrical charges to come together and thereby do something when they do. And that's exactly what happens when you're plugging something into an outlet. There is a voltage that exists, and if you connect the pathway so that current can flow through it, it will. And when you put those two prongs into the outlet, you're connecting the circuit, so now electrons can flow through the wires, and then they can actually do something. Whatever it is that you're plugging in can turn on. Well, in the same way, you have these this voltage across the membrane, and if it's allowed to come together, it can be used to do work. So, in any case, all that this means is that at rest, there is a voltage across the membrane. Okay, so if I looked at this picture, generally the way that it works is that on the outside of the there's a positive charge, and on the inside there's a negative charge. And the particles that form these charges aren't able to come together with one another. If they could, it wouldn't be voltage. Voltage means that there is positively charged stuff, and then there's negatively charged stuff, and even though they want to come together, they're separated out. They can't. And only when they are allowed to come together can they then come together. And that's the situation we have here. There is a voltage at rest across the membrane. It's the resting membrane potential. And this is what's responsible for a lot of the behavior of neurons. For example, if these are allowed to come together, the charge dissipates, and then that sends off a whole cascade of events which transfer all the way down the axon. Now, what specifically those events are, we won't get into that type of detail in this class, but it is a pretty fascinating thing that happens. So there are two things that can happen here. There can be what's known as a depolarization of the membrane, of the voltage, or there can be a repolarization of the membrane. And the reason, molecular basis for this is, again, quite complicated, but for our purposes, just know that when we say depolarization, we mean like a discharge of the battery. It's like the voltage is dissipating. If you think about neurons being like a battery, and then when they're being discharged, it would be like using up a battery that you're using for some tool. Okay, repolarization then would be like charging the battery. And neurons are constantly being depolarized, meaning they're expelling their energy, and then they're constantly being repolarized. They're constantly being charged back up. So they dissipate their voltage, they charge back up, they dissipate their voltage, they charge back up. And in the whole process, it ends up being this elaborate system that is used by the body to detect signals and transfer those signals from one place. So for example, you stimulate the receptor of a tactile, you stimulate the tactile receptor of somewhere on your skin, it will have a series of voltages that get passed from neuron to neuron to neuron to neuron, and it goes all the way up to your brain. And then you can detect it as, oh, something's touching my skin. That's a really rapid form of communication that's happening in your nervous system. Now, there is a difference between what are called graded potentials and action potentials. We said an action potential is the change in voltage that sweeps across the axon. Okay, think about it being like a gun that's firing, and it's firing a signal that goes all the way down. It's an all or nothing event. And if it happens, it happens, and it happens all the way through. And if it doesn't happen, it doesn't happen at all. That's different than what's known as a graded potential. Graded potentials are the changes in voltage experienced here at the dendrites. And again, some are stimulatory, some are inhibitory, and they will battle each other out. And if there's enough stimulatory signals to outcompete the inhibitory signals, then the molecular trigger, so to speak, is squeezed right about right here in the cell, and then that will shoot an action potential down. So these can get weaker or stronger. They can intensify or not, and then they would lead to the production of this, which is the thing that sweeps across. All right, so that's the sort of the molecular underpinning in the microanatomy of the central nervous system. It has to do a lot with these electrochemical things that are happening in these cells called neurons. And looking more macroscopically then at the anatomy, here's how the nervous system is partitioned. There is what's known as the central nervous system, and the central nervous system would be the spinal cord and the brain. Okay? Contrast that to the peripheral nervous system. The peripheral nervous system is all of the nerves that come off of either the spinal cord or the brain stem. And those would be the things that are going and innervating specific spots, whether it be specific muscles or glands or things throughout your body. Looking at the brain, specifically the anatomy here, so this is part of the central nervous system. Here's a sagittal section of what the brain looks like. Here we're seeing the spinal cord. The spinal cord is part of the central nervous system, but it's not technically considered part of the brain. Once we get through this hole in the skull that the spinal cord passes, now we're talking about the brain. And so the first part of the brain then is this thing we're seeing here. This is the brain stem. And it's one of 4 major parts of the brain. And there's additional substructures in here too, which we won't really cover all that much. Okay, this is part 1 of the brain. Another part of the brain is this little bump that you see off the back, and this is called the cerebellum. And the cerebellum is called the little brain. That's what the word means, and it's going to be something that helps with your balance and your equilibrium, your sense of positioning, and how it relates to your movements. The cerebellum is in a strategic location because it's right next to a lot of the signals that are coming in and out of the brain, so it can, in a way, monitor that activity and jump in and alter things when needed, specifically related to your positioning and your body movements. Okay, right in the middle then of your brain is a part that's called the diencephalon. And in it, there are a couple of structures, but one of the notable ones are these 2 nuggets called the thalami. You have a right and a left thalamus, and you can't really see them here because this is a sagittal section, but I'll just draw one in here. So this is about where a thalamus would sit. And the thalami are important structures because they're like relay switches. A lot of the information coming up into your brain first stops at the thalamus, and then it synapses on a bunch of neurons, which then route it out to different parts of the brain. So it's like a switching board. Then the 4th part of your brain then is this big portion which you probably generally think of when you think of a brain. This is the cerebrum. And the cerebrum is composed of a bunch of folds. You have the folds of nervous tissue. They increase the surface area. And the tops of the folds are called gyri, and the troughs, or the valleys of the folds, are called sulci. And there are different things happening in different folds. Like it's thought that the folds here are more involved in your consciousness and your reasoning and things like that. The folds here are involved in sending motor commands to your skeletal muscles, so issuing signals that go to your muscles and actually cause them to contract. Just behind that are some folds that are thought to be an important place where your sensory information is coming. So any of the touch information or the pain or the thermo information coming from receptors in your skin, it all lands here. And then in the back, this is where a lot of your visual information goes to. And so somebody that maybe slips and falls on the ice and hits their head pretty hard on the sidewalk, they could actually have some damage to their visual acuity, or maybe even partial blindness or complete blindness, depending on the severity of the injury. So each of these regions has sort of been mapped out and has a characteristic feature to it. Now, there are 2 cerebral hemispheres. There's one on the left and the one on the right. And this has spurred a lot of discussion about what it means to be either left-brained or right-brained, and I should put maybe quotes here. That it was thought that there are some people that have different proclivities. Some people are better at doing like verbal reasoning and reading. Other people are better at like 3-dimensional orientation and arrangement of objects in their mind's eye. and some people are more creative and artistic, others are more analytical and rational. It's thought that maybe your proclivity to being one way or another maybe has some roots in which side of the brain you predominantly use. I'm not entirely sure of the validity of this, but I mean it is a common conception that people have that they're either right-brained or left-brained, and some of the scientific analyses that wrought these convictions come from this gentleman here, Roger W. Sperry, and he was a psychologist studying this sort of thing. And basically what he did is he would conduct experiments in patients who had their corpus callosum severed. So the corpus callosum is this little structure here that allows the two sides of the cerebral hemispheres to talk to one another. And in these patients, it was severed. Generally, it's severed because it was some sort of treatment for epilepsy. Okay, so they'd had this treatment completed, so then now their brain was severed. And so what was happening then is that information coming from the left eye would be routed over to the right side of the brain. Information from the right eye would be routed over to the left side of the brain. And what he would do is he would show two words, project them on a screen, and then in addition to that, there would be this little surface where various objects that were representations of these words would be found. So for example, this word maybe here said key, and this word over here said maybe pen, and then there would actually be a key and a pen on there. And when asked what one of the words was on one of the sides, they could clearly say this is the word, and then when they were asked to point to it, they wouldn't be able to point to it. And then on the flip side, on the other side, if they asked what the word was, they had no recollection of what the word was. It was almost like they were blind. They couldn't see the word. But very curiously enough, if asked to point to the object that they saw, even though they couldn't tell the word that they saw, they were able to point to it. So it was like their ability to communicate language was lacking in that part of the brain, but still they were able to recognize the spatial visual characteristics of the object. And this is a summary from the paper basically saying visual material projected on the right half of the field can be described in speech and written essentially in a normal fashion. However, the same material is projected on the left half of the field, so going over to the right brain. The subject consistently insists that he didn't see anything. But the most bizarre part of it all, if asked to tell you what he saw, he can use his left hand to point to the matching picture or object. So a really interesting set of studies there done by Roger Sperry. Okay, and there's been others since too that either lend support or even maybe dispute some of these findings. But so you're maybe left-brained or right-brained, and I can't remember which one is thought to do what, but it's an interesting study nonetheless. Okay, other parts of the brain of interest, there's a pituitary gland right here, and this is one of the endocrine organs. And another thing that I'll point out is that the corpus callosum is right here, just above the diencephalon. And this is the thing that was severed in those experiments. And another unique feature of the brain is that it sits suspended in this bath of fluid, which is called cerebral spinal fluid. It's made somewhat in the middle of the brain, and then it goes out and spreads out to different parts of the brain, and this is actually continuous all the way down the spinal cord. So the fluid that accumulates here in the middle of the brain, it goes down this thing called the cerebral aqueduct, and then it passes all the way down the spinal cord, down, 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 and then from there it can get out, and now it actually bathes and nourishes the entire spinal cord around it. An interesting thing that can happen here is some women, when they are getting an epidural, which is a shot in their lower back to help numb the pain in childbirth, generally the needle doesn't go so far that it punctures the membrane that contains the cerebral spinal fluid, but occasionally it will pop through there, and then the cerebral spinal fluid starts leaking out, and so it starts actually draining the brain case, so to speak. There's a depletion in fluid, and it can lead to really excruciating headaches. And a woman would know that this is the case if she lays down and the headaches go away, and then as soon as she stands up, the headaches return. That would be an indication that it's something to do with the level of cerebrospinal fluid. And it would make one suspect that the epidural needle maybe was inserted too far and actually punctured the membrane. Then they actually have to go in and inject some blood from the mother in there, and it will coagulate and patch the hole. It's called a blood patch. But yeah, cerebrospinal fluid is very similar to blood plasma, but it has a couple of unique restrictions to it. There's actually a blood-brain barrier that make it such that not all the things in the blood can pass into the cerebrospinal fluid. Okay, now if we looked at the very surface of the cerebrum, here's all those folds that I was talking about. And what you're seeing here, this is called Brodmann's map. And this is actually the map that was created by a physiologist or histologist who was studying pieces of the brain under a microscope and looking just at the shapes of the neurons. And he would notice there were some patterns, and he actually created a map to show where some of the morphology of the neurons would change into these different regions. And this convention actually we still use mostly today. So it's amazing how accurate this was, based on the fact that it was made over 100 years ago. And that just is sort of lends credence to the idea that it's the morphology or the anatomy of a structure which often confers its function or its physiology. But in either case, a couple of unique regions here that can be found. This one right here is called the precentral gyrus, and this is going to issue motor commands. And then just behind that, this is called the postcentral gyrus, and this is going to receive sensory information from your skin. There is a region here called Brodmann's region, and that's responsible for production of language. There's the visual area here where our visual information comes from. There's auditory cortex that receives information. There's Wernicke's region that has to do with understanding language. So the part of your brain that produces language and the part of your brain that understands and comprehends language are two different parts. And here's sort of a summary of that map. And we don't have to go into too much detail here in this class, but I just want to generally show you that where we are in the cortex of the cerebrum has different functions. Okay, another interesting thing specifically about the postcentral gyrus, the place where your sensory information comes from, is that you can actually map it out, and it forms what we call a somatosensory homunculus. So it looks like this. There are, depending on where in the body the stimulus is coming from, it will get routed to a specific spot along that gyrus. So for example, if your toes were being touched, it would light up a part light up the gyrus right here in this location. If your arms were being touched, it would be generally somewhere around here. If your hand was being touched, we would detect that somewhere on this part of the gyrus, and then your face would be further down. So it's all mapped out, and that's one of the ways that your body can more or less code where the stimulus is coming from. And that's a really great thing, right? It would be somewhat sad if your body only had the ability to detect if it was being touched or not, and so it would just be like an on and off switch. But that's not our perception of tactile sensory experience is much more complicated than that. Not only can you determine if you're touched or not, you can determine where you're being touched, and you can also determine how forcefully or the level of intensity of the touch. That involves all sorts of complicated sensors scattered throughout your skin, and it all gets routed and mapped to your brain, and that is what helps to distinguish a touch, let's say, that's coming from your leg versus a touch, let's say, that's on your nose. Okay, then lastly, one little tale here. This is the tale of Phineas Gage. And Phineas Gage is somewhat of an anomaly in the medical community. His story is that he was working on the railroad, and specifically his job would be to bore holes into the rock fill them with gunpowder, and then tamp it down, and then blow it up. And that's how they would blow up big parts of rock and carve out tunnels and things for the railroad. And on one occasion, he had put the gunpowder in, and he was tamping it in with a tamping iron, which is actually what you're seeing right here. This big iron thing was put in the hole to pack down the gunpowder. And he was standing over that hole, and something happened where a spark was created or something, but it set the gunpowder ablaze, and it shot this tamping iron right through his skull, and basically blew a big hole right into his brain and out the other side. And most people would think that this would be, pretty much lights out at that point. But some coworkers brought him down to a physician. The physician cleaned the wound. He had to scrape some of the brains away and treat it and bandage it up. And Phineas Gage is reported to have said that he does not care to see his friends, as he shall be to work in a few days. So a different breed back there in the 1800s. You get a tamping iron blown through your brains, and you don't really want to see anybody because you're just going to get up and go to work the next day. Well, unfortunately for Phineas Gage, this wasn't the case. His wound got worse. It started to inflame, and it started to get infected, and there was periodically cleaning that was required. The brains would actually somewhat ooze out of the hole, and they'd have to scrape them off and treat it and bandage it. And he went unconscious for a while, but eventually did come out of it and lived a fairly normal life afterwards. I mean, I think if you read some of the reports, his friends thought maybe he wasn't quite the same, but still highly functional in a lot of respects. And sort of an interesting case study here, and the power of the brain to continue on even despite that type of level of trauma. Okay, well, that's a little survey of the nervous system, and in the next part we'll tackle actually some of the interesting facets of the mind-body problem. We as humans are very curious organisms, and I would argue quite distinct in the world, and I think a lot of the root for that gets into something that's going on with the nervous system. There's sort of a boundary there between the uniqueness that makes us human and then also the machine that is more animalistic. And this presents in a variety of ways, which we'll sort of explore, but it's more of a philosophical lecture, but at the same time, I think some really important philosophical applications and thoughts to consider to pair along with sort of the mechanistic approach that we're looking at here in this lecture. In either case, that will wrap us up then for the entire course. We're going to end on the nervous system and some of the unique unique concepts to discuss about it. And stay tuned for the next lecture.